हेलो फ्रेंड्स, आज का टॉपिक है डाइजेशन एंड एब्जॉर्प्शन का पार्ट 2। देन पार्ट 3 आएगा। इंटेस्टाइन इट इज डिवाइडेड इनटू टू पार्ट्स, स्मॉल इंटेस्टाइन एंड लार्ज इंटेस्टाइन। Small intestine is distinguishable into three regions: C-shaped duodenum, a long coiled middle portion, jejunum, and a highly coiled ileum. The opening of the stomach into the duodenum is guarded by the pyloric sphincter. For absorption and digestion food, a very large surface area is required. Therefore, some adaptations are present here. Number A, great length of the intestine. Number B, villi and microvilli. Number C, plaque circles, circularis, valves of carrying valvular conventions. Large intestine. Ileum opens into the large intestine. Large in diameter. Large intestine is differentiated into three parts: cecum, colon, and rectum. The lower end of the ileum opens on the posteromedial aspect of the ileocecal junction. The ileocecal opening is guarded by ileocecal valve. Cecum is a small blind sac which hosts some symbiotic microorganisms. About 2 cm below the ileocecal orifice, a worm-like structure emerges from the cecum called as vermiform appendix. It is a vestigial organ. Cecum is well developed in rabbit and other mammals but is vestigial in human. Colon. Cecum continues in colon, which is the middle part of large intestine. The longitudinal muscles coat from three ribbons-like bands called teniae coeli. Due to the presence of teniae, pouch-like structure develops in lumen of colon called as haustra. Colon has flowing parts in human, an ascending, a transverse descending part, and a sigmoid or pelvic colon. Rectum. This colon then continues in a uniform tube called rectum, a storage chamber for feces. Rectum opens into a small bag-like structure called anal canal. Anal canal. Anal canal open outside by anus. Anus is controlled by anal sphincter. Two types of anal sphincters are found at the opening of anus. Two types of sphincter muscles are found in anal canal: internal anal sphincter, involuntary; external anal sphincter, voluntary. Histology of alimentary canal. Unilayered epithelium in stomach, duodenum, jejunum, ileum. Mucosa. Multilayered epithelium, simple columnar स्टमक में और सिंपल कॉमनर ग्लैंडुलर ब्रश बॉर्डर्ड एपिथेलियम फॉर्म ब्लंट विलाय ड्यूडेनम में और स्केब्ज फ्रॉम लॉन्ग पॉइंटेड विलाय जेजुनम एंड इलियम में. Esophagus. Sub mucosa of esophagus and stomach, duodenum, jejunum. Branch of sympathetic and parasympathetic nerve fibers. Muscular layers. Circular. In esophagus, circular. Oblique circular stomach, circular duodenum, jejunum and ileum, circular. Nerve fiber. Esophagus and stomach, duodenum, jejunum and ileum. Branch of sympathetic or parasympathetic nerve fiber. Longitudinal. Esophagus longitudinal, stomach longitudinal, duodenum longitudinal and jejunum and ileum longitudinal. Serosa, made up of simple squamous epithelial or mesothelium. In esophagus, areolar plus WFCT called tunica adventitia. In stomach, SSE plus fats plus lymph tissue is equal to omentum. Duodenum serosa, jejunum and ileum serosa. Wall of alimentary canal is made up of four layers, outer to inner. Number first, serosa. It is outermost layer of gut. It is called tunica adventitia in esophagus. Serosa is made up of a thin mesothelium, visceral peritoneum with some connective tissue. Tunica adventitia is made up of white fibrous connective tissue and areolar connective tissue. Number two, muscle layer. It is formed by circular inner layer and longitudinal outer layer of smooth muscles. Thickest layer is found in stomach, maximum peristalsis, and thinnest layer in rectum, minimum peristalsis. Number third, submucosa. It is loose connective tissue layer with blood vessels, lymph vessels, and nerves. Number four, mucosa. It is the innermost layer of gut which contains the secretory and absorptive cells. It is differentiated into three parts. Outer part, called mucosa muscularis. It is made up of longitudinal and circular muscles. It is important role in exposing of surface area for the absorption. They also provide support to the folds of alimentary canal. Middle part, it is called animal propria. It contains few modified lymphatic tissue which provides immunity. Example, Peyer's patch. It is made up of reticulate and fibrous connective tissue. Innermost part called muscular layer. In esophagus, this layer is made up of non-keratinized stratified squamous epithelial. Except esophagus, thin layer is single layer thick, which is made up of columnar mucous epithelium. This layer makes the lining of lumen of alimentary canal. This layer makes the fold of alimentary canal. Folds of esophagus are less developed, whereas fold of stomach in finger shaped and develop as gland called gastric gland. Folds of small intestine are conical shaped called villi. A small slit-like space is found at the base of villi. These spaces are called crypts of liver cone. Villi of duodenum are small blunt. Villi of jejunum and ileum are long and pointed. Maximum villi are found in jejunum. Diagrammatic representation of transverse section of gut. Serosa. Inner, circular, outer, longitudinal, submucosa, mucosa, lumen, muscularis. Werner's gland. They are small spherical multicellular glands. They open into crypts of liver cone with the help of fine tubules. These glands are found in the submucosa of duodenum. They synthesize and secrete the non-enzymatic alkaline secretion of interstitial juice. Penith cells. Number first, these cells are found in mucosal layers of shifts of liver cone of jejunum. Number second, they are unicellular glands. Number third, they synthesize and secrete lysosome and diaphragm. Number four, the secretory substance of Werner's gland and crypts of liver cone are combinedly called intestinal juice or succus intricus. Peyer's patch. They are small lymph nodes which are found with the mucosa of small intestine, jejunum, and ileum more in number. They are also called intestinal tonsils and provide immunity. Nerve supply. Two types of nerve plexus are found in muscle of alimentary canal. Orvax nerve plexus, menintric plexus. This nerve plexus is found between longitudinal muscles and circular muscles. It controls muscles contraction. Meissner's nerve plexus found between circular muscles and submucosa, but in stomach it is found between oblique muscles and submucosa. Digestive gland. The digestive gland associated with the alimentary canal include the salivary glands, the liver, and the pancreas. Salivary gland. Saliva is mainly produced by three pairs of salivary glands: the parotid cheek, the maxillary submandibular lower jaw, and the sublinguals below the tongue. These glands are situated outside the buccal cavity, secrete salivary juice into the buccal cavity. In humans, three pairs of salivary glands are present. Salivary glands, three pairs: parotid, submaxillary, sublingual. Location: below ear cheek, submaxillary, jaw angle, low jaw, sublingual, below tongue. Number: one pair, one pair, one pair. Duct: extensions duct, Watten's duct, duct of Ravinus or Bartholin's duct. Secretion: saliva plus enzyme, saliva. Special point: largest salivary gland and viral infection cause mumps. Secrete maximum amount of saliva. Neet. Composition of saliva. Salivary glands are not present in whale and frog. Important. Water 99.5%, mucus, starch, digesting, taline, enzyme, lysozyme, and thiocyanins, and few ions like sodium, potassium, chlorine, IgA, antibody, urea, and uric acid, etc. are present. Taline is secreted mainly by the parotid gland, lysozyme, and thiocyanins mainly kill bacteria. They also check the growth of bacteria in buccopharyngeal cavity. Salivation is stimulated by cranial nerve number 7 and 9. Sympathetic nerve system decrease the secretion of saliva, while parasympathetic nerve system increase the secretion of saliva. Liver, falciform ligament, diaphragm, gallbladder, duodenum, ascending duct, accessory duct, body, tail, pancreatic duct, common hepatic duct, common bile duct, left hepatic duct. It develops from endoderm, weight 1.2 to 1.5 kg. In human, it is found in right side of abdominal cavity below the diaphragm. The liver is the largest gland of body. It is made up of two lobes, left and right lobe. Left lobe is smaller than right lobe. Right lobe from 5/6 of the liver, and left lobe from 1/6 of liver. Right and left liver lobes are separated from each other by the falciform ligament, fibrous connective tissue, which is made up of fold peritoneum. Right and left hepatic duct develop from right and left liver lobe. Both these ducts combine to form a common hepatic duct. The hepatic lobules are the structural and functional unit of liver, containing hepatic cells arranged in the form of cords. Each lobule is covered by a thin connective tissue sheath called the Glisson's capsules. The bile secreted by the hepatic cells passes through the hepatic ducts and is stored and concentrated in a thin muscular sac called the gallbladder. The duct of gallbladder, cystic duct, along with the hepatic duct from the liver forms the common bile duct. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by a sphincter and called the sphincter of Oddi. Gallbladder. Gallbladder is situated below right lobe of liver. Gallbladder is not present in lamprey, whale, and horse. Important. Cystic duct of gallbladder is connected to common hepatic duct form a common bile duct, also called ductus coledox. Functions of liver. Liver is known as biological and chemical factory of the body. Most of the biochemical functions of the body are done by the liver. Number first, secretion and synthesis of bile. This is the main function of liver. Bile is yellowish green alkaline fluid in bile juice, bile salt, sodium bicarbonate, glycocolate, taurocolate, bile pigment, cholesterol, lecithin, etc. are present. Bile salt help in emulsification of fats. Bile prevent the food from decomposition. It kill the harmful bacteria. Number 2, carbohydrate metabolism. The main center of carbohydrate metabolism is liver. Following steps are related with carbohydrate metabolism. Glycogenesis. The conversion and storage of extra amount of glucose into glycogen from the digested food is called glycogenesis. The main store food in the liver in glycogen. Glycogenolysis. The conversion of glycogen into glucose. And when glucose level in blood falls down is called glycogenolysis. Gluconeogenesis. At the time of need, liver converts non-carbohydrate compounds, for example, amino acid, fatty acid into glucose. This conversion is called gluconeogenesis. This is the neoformative process of glucose. Glyconeogenesis, synthesis of glucose from acid which comes from muscles, is called glyconeogenesis. This is the neoformative process of glucose. Number 3, storage of fats. Liver store fats in a small amount. Hepatic cells play an important role in fat of metabolism. The storage of fat increase in the liver of alcohol acidic persons, fatty liver. This stored fat decrease the activity of liver. The damage of liver due to alcohol intake is called alcoholic liver cirrhosis. Number 4, deamination and urea formation. Deamination of amino acid is mainly done by liver. Amino acid NH3. Liver converts ammonia, more toxic, into urea, less toxic, through ornithine cycle. The spoilage of liver, the ammonia level is the animal body increase and ultimately the animal dies. Number 5, purification of blood. Kupffer cells of liver and sphenocytes of spleen are the phagocytic cells, help in phagocytosis of dead blood cells and bacteria from the blood. Number 6, synthesis of plasma proteins. Many types of proteins are present in blood plasma. All the proteins except gamma globulins are synthesized in the liver. Chemically, antibodies are gamma globulin formed by lymphocytes. Number 7, synthesis of heparin. Heparin is a natural anticoagulant mucopolysaccharide. Some heparin is also formed by basophils, granulated WBC and mast cells. Number 8, synthesis of vitamin A. The liver changes beta carotene into vitamin A. Beta carotene is a photosynthetic pigment which is obtained from yellow part of fruit. It is abundantly found is correct. Number 9, liver store vitamin A, D, E, K, B12. Number 10, storage of minerals. Liver store iron, copper, zinc, cobalt, molybdenum, etc. Liver is a good source of iron. Number 11, hemopoiesis. The formation of blood cells is called hemopoiesis. In embryonic stage, RBC and WBC are formed by liver. Number 12, secretion of enzyme. Some enzymes are secreted by liver, participate in metabolism of proteins, fats and carbohydrates, for example dehydrogenase, cytochrome oxidase, etc. Number 13, prothrombin and fibrinogen proteins are also formed in hepatic cells. These help in blood clotting. Number 14, factors 1st, 2nd, 5th, 7th, 9th and 10th are formed in liver which are responsible for blood clotting. Deficiency of selenium cause liver necrosis. Bile juice. In the middle part of the duodenum, bile juice is secreted. The parenchyma cells of the liver produce bile juice, and it is stored in the gallbladder. Bile juice does not contain any digestive enzyme, therefore, it is not a true digestive juice. Pseudo digestive juice. Composition of bile juice. Bile juice is a greenish, biliverdine, yellow, bilirubin colored alkaline fluid. Composition of liver bile. pH 8.0, water 98%. Organic constituents are bile acid, bile pigment, cholesterol, lecithin. Inorganic constituents sodium, potassium, etc. Bile pigments are the excretory substance of the liver. Bile salts are of two types. Number first, inorganic salt. Bile juice contains sodium chloride, sodium Na2CO3, NaHCO3, etc. in it. Inorganic salt neutralize the acidity of the food and makes the medium basic. It is necessary for the medium to become basic because the pancreatic juice enzyme can act only in basic medium. Number B, organic salt. Organic salt like Na-glycolate and Na-taurocolate are found in bile juice. The main function of these salt is the emulsification of fats because pancreatic lipase and act only on emulsified fats. Bile salt also help in the absorption of fats and fat-soluble vitamins A, D, E, K. Bile salt combine with fats, cholesterol, phospholipid, lecithin, and these vitamins to form compounds called micelles, which is absorbed rapidly in the form of micelles. Cholesterol and phospholipid lecithin remain soluble. Function of bile juice. Neutralization of HCl. Its sodium neutralizes HCl of chyme semi-fluid food found in the stomach. Emulsification. Sodium glycolate and sodium taurocolate are bile salts which break the large fat droplets into the smaller ones. Absorption of fat and fat-soluble vitamins. Its salts help in the absorption of fat and fatty acid and glycerol and fat-soluble vitamins A, D, E, and K. Excretion. Bile pigments, bilirubin, biliverdin, and excretory products. Prevention of decomposition. Bile is alkaline, hence it prevents the decomposition of food, preventing the growth of bacteria on it. Stimulation of peristalsis. Bile increases peristalsis of the intestine. Activation of lipase. Bile contains no enzyme but activates the enzyme lipase. Bile pigments, cholesterol, and lecithin are the secretory substance found in bile juice. Gallstone. Sometimes the passage inside the bile duct gets blocked or become narrow, so the cholesterol gets deposited or precipitated in the gallbladder. This is termed as the gallstone cholestasis. Obstructive jaundice. If the passage of bile is blocked, then the amount of bilirubin increase in the food, so the yellowish coloration of body like skin, cornea, and nails appear yellow. Urine also become yellow. Pancreas. Sweet bread. It develops from endoderm, which is soft, lobulated, and elongated organ. It is divided into head, neck, body, and tail regions. It is made up of numerous acini. Acini is a group of secretory cells surrounding a cavity. Each acini is lined by pyramidal shaped cells. Pyramidal shapes. These acinar cells secrete the enzyme of pancreatic juice. Each acini into pancreatic ductal. Many pancreatic ducts combine to form main pancreatic duct of Warshang. The main pancreatic duct is joined with the bile duct to form the hepatopancreatic ampulla, which opens into duodenum. The accessory pancreatic duct of Santorini opens into duodenum with separate opening located above the opening of major pancreatic duct. Some group of endocrine cells are also found in between group of acini called islets of Langerhans. These islets secrete insulin and glucagon hormones, so this gland is exocrine as well as endocrine. Heterocrine, its 99% part is exocrine while 1% part is endocrine. In human, both bile duct and pancreatic duct combine to form common duct called as hepatopancreatic duct. The terminal end of common duct is swollen and is called a ampulla of Vater or hepatopancreatic ampulla. Ampulla of Vater opens into middle part of duodenum and is controlled by sphincter of Oddi, while bile duct is controlled by sphincter of Boyden. Pancreatic juice. Pancreozymin stimulates the acini and glandular cells, so pancreatic juice is secreted. The pancreatic juice is secreted by the exocrine cells of the pancreas. Pancreatic juice is highly odoriferous, colorless, basic fluid which contains enzymes and salts. Composition of pancreatic juice. Total amount in men is equal to 1 liter to 1.5 liter per day. Water 98%, pH 7.5 to 8.3, salts 2%. Pancreatic juice contain only inorganic salts.